At the very beginning of the lecture, we've brainstormed quite a bit about eugenics. I've given an introductory spiel about it. I think there's some common modes. And here I thought of a few. I don't remember how many I thought of. But if you can think of any more, please let me know. We can add it to the list. So I think a very common mode is prenatal screening. I mean, this does occur. By the way, I want to emphasize that we've tended now to talk about eugenics quite negatively, and I think for good reason. However, maybe some modes of eugenics or eugenics under certain circumstances are morally permitted and perhaps even mandated under certain normative ethical theories. Okay, so the first is prenatal screening. This occurs all the time. From my understanding, there are really two ways to do it, but in the 8 weeks, what happens is you get a sample from the placenta, and then you do, well, you get a sample from the placenta, you get the child's DNA out, and then what you do is not map the whole entire genome. That'll be incredibly, well, not too expensive nowadays, it's quick. But you do PCRs just to figure out if there are certain genetic markers for, let's say, 200 different diseases. What diseases? Down syndrome, Alzheimer's, Huntington's, and so on and so forth. And so you'll get a readout about the probability that the child would have a disease. I mean, with Down syndrome, you can these are objective probabilities, not epistemic probabilities. So with Down syndrome, just by genetics alone, you can definitely tell if there's Down syndromes, but with things like Alzheimer's, you won't know. In fact, you won't know until advanced age. But they do give you, let's say, 20% that you get Alzheimer's, which I don't know how it compares with the population norm. Okay. We've got population level mating control. This is what I've called it. So I mean, all these things, I've made these terms up. What I mean, population level mating control. What I mean is, there are all these different types of people in a population. You don't let certain kinds reproduce with other kinds. In fact, you don't let certain kinds reproduce at all. So perhaps people who have certain illnesses or certain characteristics are not allowed to reproduce, and over time, those characteristics, the thought is that they'll disappear from the population, although that's really not how genetics works. It does depend on the disease, though. Okay, we've got genetic alterations, which is something that was mentioned at the brainstorm and intermittently throughout the chat. We've got various technologies. The latest one is CRISPR. Now, CRISPR, I forgot what it stands for, but it's a very interesting technique. I think it was taken from either a bacterium or a virus, and then adapted for our own use. So what happens is you can create this sort of molecular or cellular honing device that starts going into, well, you inject it into cell, and then what happens is it finds the DNA, it scans the double helix, and then it finds the right part, and then it splits it. Or suppose you want to split it first. You find something else to split it, you find some muscle, basically it's analogous to real life, right? You want something done, you need some guy with muscle, you need some guy who scans the scout, and then you need some guy, the engineer, who does the alteration. So first you've got this muscle guy, comes in, splits the double helix into the 2 strands, and then you've got another guy that comes, scans the double helix, finds the right sequences, right? ATG, ATGA, whatever, right? Whatever you want. I mean, obviously it's going to be much longer than that. But you find the right sequence, And then you've got another guy which is designed to specifically replace that sequence of DNA with another one, suppose GGGA or something. Okay. That's the idea. It literally goes in there, opens up the double helix, scans for the right part, and then replaces it with what you want. Okay. Of course, you're going to, I mean, most of these technologies are going to occur in a germ cell. You're not going to, you're not going to alter the DNA of a skin cell. That's useless. It's going to die off pretty soon, especially, well, maybe not a skin, but something deeper than the skin. It's going to die off pretty soon. So you want to do it to, say, a gamete, a sperm or something, or an embryo. Okay. That's CRISPR. There's a question. The government prohibiting inbreeding is a population-level mating control. I think so, actually. I do believe that. So inbreeding as in some incestuous sexual relationship, that sires offspring. I mean, that's illegal in a lot of countries. That's eugenics as well. And I think actually most people would think that's okay, right? That kind of population level mating control is okay. I think people get quite disgusted by this. And I actually think for innate reasons rather than cultural reasons. There's actually a lot of good evolutionary literature on this. But of course, as we know, inbreeding results in a lot of genetic defects. And this is one of the reasons why the government presumably prohibits it. Okay, there's population level existence control. Aha, the key word is existence. So they control who exists and who doesn't. If you don't exist, you don't get to mate. With existence, I don't know if indirect measures count as well. So it's not the case that you have to directly kill someone to control their existence, but quite indirect measures like not allowing them to get a job or making it harder for them to get a job or doing other certain circumstantial things to them. I mean, that's quite indirectly controlling their existence over time. Okay. Oh, I just have 4. Can anybody think of more that don't slot into any of these 4? Are we missing anything? I mean, genetic alteration, that's quite broad. Can we have postnatal screening? Okay, what about harvesting the sex organ? You mean sterilization? Ah, okay. Yeah, okay. That's something that I failed to mention. So I don't think people harvest them. I mean, harvest them to sell, trade. So I don't think that happens. I think of species, yeah, but not humans, of animal species. So in mating control, I think we should put sterilization as well. I mean, this happened in Nazi Germany. And I'm pretty sure it's happening today. Okay. What do I have here? Okay, suppose we have an event. I don't even know why I call it event group, but suppose we have some event or type of event. I think I know why now. Okay, suppose we have a category of events, right? And these can be interspersed through time. We've got E1, E2, E3, E4, whatever. the aggregation of these events determine which types of people get born. These events, individual events, are really just birth events. So E1 is, say, birth of a T individual. E2 is a birth of a T individual. E3 is the birth of an R individual. If you have 10,000 million of these events, then you get some population level statistic about which types of people get born. Maybe it's 80% T and then 10% R, whatever, okay? What factors go into these types of events? Well, I think prenatal screening is one factor. Here's what I mean. Suppose, we've got prenatal screening and we've got a mother and a father. There's some prenatal screening performed on the child. They see that the child has Down syndrome. And suppose they then abort I shouldn't say child yet. Suppose they abort the embryo. Then what happens is there's going to be less people of that type, namely that have Down syndrome, that gets born, right? At least one. If lots of people do this, then there'll be maybe more. So what this diagram shows is prenatal screening is influencing which types of people get born. Okay? Does everyone follow? Okay. Now, the point of this also is that prenatal screening occurs immediately in the causal chain before the birthing events. We may have others that lie at different points of the causal chain. For example, who gets to reproduce with whom? This doesn't occur before the prenatal screening. Sorry, this doesn't occur after the prenatal screening, but before it. So it affects which types of people get born, but before any prenatal screening could occur. So all of these events are supposed to be eugenic events. So this is eugenic, and this is also eugenic. Of course, this most of the time is voluntary, right? Who gets to reproduce with whom? As I said at the very beginning, it's quite natural for you to be picky about your date, about who you marry, and about who you have children with. And I'm sure even some of those times, people are choosing on the basis of disease, but not always. Oh, yes, and I also wrote here, who gets to reproduce with whom is often a function of who gets to marry whom or with whom. Although, of course, this is a watertight. Of course, you can reproduce without getting married. Happens all the time. Not necessarily a bad thing. Some people don't want to get married. One way of also determining who gets to reproduce with whom is determining physically where people are. If you lock them up in a cell, they're not going to be able to reproduce with lots of people. Only the people in the cell, if they're locked alone, then they've got no one to reproduce with at all. Of course, the determination of the people who get to live is also one way of determining who gets to reproduce with whom. My point here is all of these things are united by the fact that they ultimately affect these types of events through reproductive facts. Okay, I think that's right. However, genetic alterations, stuff like CRISPR, I think they, I mean, maybe this should be on the same level as prenatal screening. For the reason that it seems to occur after who gets to reproduce with whom. So, suppose you have John and you have Susie, they reproduce, then we've got embryo, and then maybe you do some genetic alterations to the embryo. Well, I actually don't think genetic alterations have to occur in the embryo. I think it can occur for the gametes. Which brings me to the question of, okay, who gets to reproduce with whom? This is still quite coarse-grained. It's got various intermediary steps within it. I mean, what exactly do you mean by reproduce? Do you mean sexual intercourse? Do you mean fertilization? And so on and so forth. Okay, good. I thought this was quite a neat diagram. But are there any questions or thoughts or concerns so far? Are we missing anything? Is something not at the right level? I think one thing I want to say is, maybe I was slightly inconsistent with this. I don't mean to say that they all go through prenatal screening. Of course, sometimes people reproduce with others and then there's no prenatal screening occurring. Okay, in that case, let's move on. Okay, so I want to go a bit more detail into prenatal screening. I think I've talked a bit about this already, but most of the time, you're really just looking at DNA. You're really just looking at the genetics. Why did I say morphological here? So it's not necessarily morphological, but you're looking for some sort of signal. In this case, it could be certain genetic markers after performing a PCR. Prenatal screening can be split into, I think, two types. You can have some positive ones, all right, which is once you screen them, you're now saying, okay, you get to continue to the next stage. You get to be a contender for fertilization. I mean, this happens in IVF treatments all the time. The thing with IVF treatments is usually you're not just picking, well, let's say, well, you have multiple eggs going on at the same time usually as well. You don't have just one sperm, one egg. You have a few eggs from the mother, and then you have a lot of sperm from the father. And you try on multiple occasions to fertilize them and then implant them into the body. Now, a lot of the fertilization fails, so it's really pragmatic to do multiple times at once, right? You don't put all your eggs in one basket, no pun intended. But in terms of IVF, you can often screen the gametes. So you can screen the sperm. You have so many sperm to pick from, and you can only, say, work with 10 sperm. So what do? You pick a bunch and you just screen them and make sure that 10 that you're using are going to be up to par to some standard, namely that it does not have any of the major diseases. It would be a shame if you spent all that money, all that time, and you fertilize an egg with a sperm that has quite a severe disease. The thought is it's a shame because it's a shame for the mother, it's unfortunate, but also for the child as well. But we have prenatal screening that is negative, which is not the case that, look, you're screening from sperm and you're saying, okay, you get to make it to the next stage. What you're doing is you're literally removing it from the reproductive cycle. You're terminating the embryo, terminating the fetus upon deducing or observing certain markers. This happens quite a bit. It's legal in most countries. And ethically speaking, I mean, we discussed this before, but it seems to be the case that most people think ethically it's okay. It really depends on the disease, doesn't it? I mean, what if it's quite a severe disease? That's not going to be good for the child if they were to be born, and it doesn't seem good for the mother either. Okay, of course, not all diseases are genetic or hereditary. It's just somewhat developmental. This is just to say that you can't screen everything. I mean, some diseases don't. You don't get some diseases until after you're born. But also, yeah, I suppose that if they're developmental, they can be screened as well through other ways, not through genetic markers. I mean, one thing is just looking at morphological features. Suppose you're doing an ultrasound and you see that the baby has one more leg. I mean, there you go, you've screened. So when we're screening, you tend to screen gametes on their own, well, for IVF at least. You tend to screen gametes on their own, but in some cases, there's actually no disease there or no disease by itself, but when it's combined with certain other genetics, then a disease may arise. I mean, there are lots of things to be said about prenatal screening. If you're interested in the ethics of prenatal screening, which may turn on some of the details, feel free to do research into it. I now want to make a distinction, and I think this is an old distinction. What I mean by old is that it's existed for some time. The distinction is between positive and negative eugenics. So I suppose Galton lived before J.B.S. Haldane. But J.B.S. Haldane is I suppose Galton is an evolutionary biologist in his own right. I believe he's the cousin of Charles Darwin, but he's probably known for his statistics more. Now, J.B.S. Haldane was a very, very famous evolutionary biologist. I actually think they should make a movie on him because he had a very interesting life. I mean, they made movies about Einstein, Newton, even about Charles Darwin, about Wittgenstein, but they haven't made one about Haldane yet. But he's really had a remarkable life, and I think he was born in quite a upper-class aristocratic family. I think, although they're military, but I mean, he lived from, I don't know, 1890, 1880 to 1950. So he died maybe only 50, 60 years ago. But towards the end of his life, he decided to live in India, and he's very much against the British government for various things. And I think he died in India. Anyways, his life is interesting for many reasons other than that. Now, so either him or Galton made a distinction between positive eugenics and what's called negative eugenics. So here's I think the primary difference. So positive eugenics aims to increase some trait or characteristic in a person, group, or population. So the aim is to increase the desired characteristic. But in negative eugenics, it's an enterprise that aims to reduce undesirable characteristics. So here's the difference. One's aimed at increasing desired characteristics, and the other is aimed at reducing undesirable traits and characteristics. We've encountered both of these different forms of eugenics in our lecture so far. One example of negative eugenics is killing people, right? Termination of embryos. But positive eugenics is really restricting who gets to mate with whom. Most genetic alterations are going to be positive. But of course, here I've said that, I mean, there might be some cases of genetic alterations that are negative in that they aim at reducing undesirable traits and characteristics. I suppose if you're removing some DNA sequence, then that's removing some undesirable characteristic. But at the same time, you're inserting DNA sequences. So maybe genetic alterations fall into both. And that's fine. I don't think distinctions always have to be mutually exclusive. I mean, you can say there's a distinction between a blue color and a red color, but some colors might be both red and blue. That's perfectly fine. It doesn't challenge logic. Okay, so we have this distinction, and you might be tempted to think, okay, positive eugenics is okay. Maybe I should change this color. You might be tempted to think, okay, positive genetics is okay. Look at the examples. But then you see here killing people, termination of embryo. You might think that this is bad. This is morally not okay. You might be tempted to think that. But I don't think that'll be right. I don't think moral status really tracks along this dimension. In other words, there's no covariance. Things that are positive eugenics don't seem to be always morally permissible. And things that are negative eugenics don't always seem to be morally reprehensible. In fact, I think there are instances of positive eugenics that are morally reprehensible and instances of negative eugenics that are morally permissible.